Hello everyone. Today we will discuss about denaturation of proteins. Denaturation is defined as the disruption of secondary, tertiary and quaternary organization of a protein molecule due to cleavage of non-covalent bonds. In this, primary structure of protein molecule, that is peptide bond, is not affected. The conformation of a protein molecule is extremely sensitive to changes in their environment. In denaturation, there is a disruption of native or biologically active protein conformation when the environment is altered. The average free energy of denaturation is about 12 plus or minus 5 kilocalorie per mole. This is equivalent to the disruption of 3 or 4 hydrogen bonds. Therefore, proteins are characterized by a narrow range of thermodynamic stability. Next, we will see agents that cause denaturation. There are physical and chemical agents which cause denaturation of proteins. Physical and chemical agents. Physical agents include heat, UV light, ultrasound, high pressure and violent shaking. Chemical agents are organic solvents, acids or alkalis, urea and various detergents. Next, we will see alterations in protein molecules after denaturation. Alterations of protein molecules after denaturation. The disruption of the protein molecule results in alteration of the chemical, physical and biological characteristics of the protein. Chemical, physical and biological characteristics. First, we will see chemical alterations. Chemical alterations greatly decrease the solubility, especially at isoelectric pH of the protein. Decrease the solubility at isoelectric pH of the protein. Then maximum precipitation as flocules occur at isoelectric pH of the protein. Flocules, that is maximum precipitation at isoelectric pH of the protein. Next one is many chemical groups which were inactive become exposed. For example, SH groups, they become exposed. They were inactive and become exposed. Then denaturation can be reversible. These are the chemical alterations. Next, we will see physical alterations. Physical alterations confers increase the viscosity of the solution. Then rate of diffusion of the protein molecule decreases. These are the physical alterations. Then biological alterations are increase the digestibility by proteolytic enzymes. Increase the digestibility. Biological. Increase the digestibility by proteolytic enzymes. This is found in the case of certain denatured proteins. Denaturation destroys enzymal and hormonal activity. Biologically, they become inactive. These are the chemical, physical and biological alterations of protein molecules on denaturation. Next, we will see a relationship of denaturation, flocculation and coagulum formation. Relationship of denaturation, flocculation and coagulum formation. We will take an example of albumin, behavior of denatured albumin. Behavior of denatured albumin. In the case of albumin, the denatured albumin is soluble in extremes of pH. In acidic pH, that is pH less than isoelectric pH, this is denatured protein, this is native protein. If this is native protein, it is soluble and it becomes denatured upon heat, then it is also soluble. Then in the case of alkaline pH, PR minus, denatured, it is soluble Then upon alkali, that is OH minus, it becomes denatured. This denatured protein is denatured protein undergo flocculation and forms floccules, and this is reversible. Here alkali added, OH minus. Then this this denatured protein then converts to coagulum. Coagulum formation upon heat. This is insoluble and irreversible. So in the case of albumin, denatured albumin is soluble in extremes of pH. In extremes of pH, it is soluble. Then maximum precipitation occurs at, here it is pH equals Pi. So maximum precipitation occurs at Pi of the protein, that is isoelectric pH of the protein. That is, in that case, albumin exists as floccules, that is flocculation. Denatured protein as floccules is reversible and it is soluble in extremes of pH. When the floccules at isoelectric pH is further heated, it becomes dense coagulum. This step is irreversible and is not soluble in extremes of pH. That is about behavior of denatured albumin. Next we will see reversibility and irreversibility of denatured protein. Reversibility and irreversibility of denatured protein. First we will see reversibility. Reversibility. For example, reversibility or controlled denaturation is shown by treatment of protein ribonuclease with urea and beta mercaptoethanol. With urea, beta mercaptoethanol. So, example of reversibility or controlled denaturation. Reversibility or controlled denaturation. It is shown by treatment of protein ribonuclease with the urea and beta mercaptoethanol. When the disulfide bridges are broken, the polypeptide chain uncoils. On slow reoxidation of the denatured protein without the urea, protein is converted into original native protein with original tertiary conformation. That is reversibility. Next we will see irreversibility. An example of irreversible or uncontrolled denaturation occurs in the boiling of an egg. In this case, the tertiary structure of the protein is irreversibly destroyed with the formation of a disorganized mass of polypeptide chains. Tertiary structure of the protein is irreversibly destroyed with the formation of a disorganized mass of polypeptide chains. That is irreversibility. Next is criteria of purity of proteins. Criteria of purity of proteins. First one is solubility curve. Purity or homogeneity of an isolated protein can be tested by various methods. First we will see solubility curve. Solubility curve. So we will see criteria of purity of proteins. First one is solubility curve. This is purity of proteins by solubility. When the solute is added repeatedly in increasing quantity to the solvent, the solute in solution rises proportionately and produces a sloping straight line. A sloping straight line in its solubility curve. When the solute is added repeatedly in increasing quantity to the solvent, the solute in solution rises proportionately and it produces a sloping straight line in its solubility curve. When the solution gets saturated, it fails to hold the solute in solution. So a single sharp break occurs in straight line at saturation point. These are the saturation points. More than one break in the line, there is more than one break in the line or a non-linear curve indicates non-homogeneity of the sample, otherwise heterogeneous solute. This is solubility curve. Next one is, so we are discussing about the criteria of purity of protein. First one is solubility curve, second one is molecular weight. Molecular weight is determined by electrophoresis, gel filtration or ultracentrifugation. This indicates the purity of protein. Next one is ultracentrifugation. Analytical ultracentrifugation gives a sharp moving boundary between the pure solvent and solute containing layer. So first one is solubility curve, second is molecular weight, third one is ultracentrifugation, next is electrophoresis. Electrophoretic mobility and a homogeneous sharp level. This indicates that protein is pure. Electrophoretic mobility and homogeneous sharp level. That indicates the protein is pure. Next one is chromatography. The particles of a pure protein emerge out of the chromatography column as one single peak of element. The particles of a pure protein emerge out of the chromatography column as one single peak of element. Then next is immunoreactivity. The antibodies raised against a pure protein will show only one sharp spike on immunodiffusion or immunoelectrophoresis. These are the criteria of purity of proteins. So this is about denaturation of proteins. Thank you for watching.